So what happens if something goes wrong during this process of meiosis? We see a failure of the homologous chromosomes or the sister chromatids separating properly in either anaphase 1 or anaphase 2. We're going to call this non-disjunction, okay? So normally, if this is a normal mitotic division and everything goes to plan, we're going to start with a haploid cell. We're going to end up with 4 diploid cells. If we have a non-disjunction during meiosis 1, what we're going to see is that instead of the spindle fibers attaching to each of the homologous chromosomes, we're going to see a spindle fiber attached to both homologous chromosomes, and that's going to create cells that have too much and too little genetic material. In anaphase 2, if the spindle fibers are attached to both of the sister chromatids, it's going to yoink them way over here. And we're going to end up with one cell that has too much genetic material, one cell that has not enough, and then we're going to end up with 2 cells that have the right amount. So we can look at the outcome of these gametes and determine where the non-disjunction event occurred. Okay. When we have too much or too little genetic material, we call this aneuploidy. Okay. So most of the time, gametes that have too little or too much genetic material are not viable, and any fertilization event that comes from one of those types of cells is usually not functional in that zygote isn't formed, doesn't mature, does not undergo mitosis. In humans, we can have extra DNA, an extra chromosome. So Down syndrome is an excellent example. Here, these individuals have a 3rd chromosome at chromosome number 21. Okay. This is due to one of those non-disjunction events, and an egg was fertilized that had an extra number 21 chromosome.